All good?
Great. I'll let you take it away.
Okay.
Let me hop on
Let
out.
me grab the screen here. Let's see. Let me steal the screen and see what we got. All right. Well, we're going to begin with one of the greats. We're going to begin with Lillian Hellman. Lillian Hellman was a famously hard-living playwright, a fabulously creative playwright, who had a remarkably rich life. You could tell just by looking at her what her life was like. She was blacklisted by the House Un-American Activities Committee in the 1950s and really suffered under that greatly. But in writing her autobiography, she came up with a line that I think describes the approach to evolution that underlies my entire book and many of the major transitions we see in the history of life. She said, in describing her own life, she said, "Nothing, of course, ever begins when you think it does." Such a powerful statement in so many ways, because it applies to fossils, it applies to genes, and yes, of course, it applies to Lillian Hellman's life as well. I'll give you an example. If you think that lungs evolved to help animals live on land, that feathers evolved to help animals fly, and invention after invention evolved to make great revolutions, you'd be in very good company. You'd also be entirely wrong. And we've known this for over a century. It's pretty remarkable. I'm going to work that through. But some of the greatest steps we see in this are relating to DNA and how DNA works to build bodies and how it evolves. And I'm going to show you some of the surprises. I'm going to give you with one of these surprises to show that nothing ever begins when you think it does. So DNA, we have DNA in every cell of our bodies. We have about 4 to 5 trillion cells inside of us. And inside of each one of those cells, inside of each nucleus of each cell, is a 6-foot-long strand of DNA. Think about that. A 6-foot-long strand of DNA is rolled, packed, and crumbled in on itself to fit inside each of every one of those 4 to 5 trillion cells. There's so much DNA inside of us that if you were to take all the DNA in our bodies and lie it end to end, it would extend from here almost to the planet Pluto. That's a lot of genetic computing power. And what we've done as scientists over the last 2 decades is sequence so many different genomes. You know, we started with the Human Genome Project, but we now have genomes for so many species, thousands upon thousands of species. We can now sequence a genome in an afternoon for about $1,000. I mean, it's truly remarkable. And when we look at that genome, we find incredible surprises. And let me just give you one surprise. And it relates to a colleague, a professor and neurobiologist, Professor Jason Shepard at the University of Utah. Jason doesn't work on DNA per se, but he works on memories. He's a neurobiologist. And he was interested in studying a gene that's involved in memories in people and mice and other animals. And this gene is called ARC, A-R-C. So he's studying this gene ARC, and the reason why is because mice that have a mutation in this gene ARC lose memories. They can solve a puzzle during the day, but the next day they can't remember their solution at all. Likewise, humans that have mutations in the gene ARC also have a variety of cognitive deficits. So he's studying ARC. And what he does is he takes the protein that ARC's made and he pops it under a microscope. So he's kind of looking at the product of the gene ARC, pops it under the microscope and takes a peek at it. And he looks at this, here it is. Doesn't look too remarkable. Look at the slide there and you see these little blebs in there, right? He looks at those, he says, you know, I've seen these before. Scratches his head. I know where I've seen them before. I've seen them, these little blebs in other things. Looks exactly like the blebs made by the protein, the gene that causes AIDS, HIV, the virus that causes AIDS, HIV. So wait a minute, a memory gene in mice and people looks a lot like a virus, the clumps that are made by a virus, such as HIV, the virus that causes AIDS. So he's at a medical school, so he calls a colleague in the building next door who works on HIV, and invites them over to the lab and shows them the slide and doesn't tell them what's on the slide. Says, "What do you see?" And the HIV expert says, "Oh, of course I'm looking at HIV." Wrong. ARC, a memory gene. How's that possible? Well, they looked at the structure of ARC and they compared it to the structure of HIV, and it turns out a gene that makes memory in mice and people and other animals is actually a modified virus. It's a repurposed virus. The idea is once they sequenced all these different genes and compared them to among different species, they found that the hypothesis that at some point in the distant past, in one of our distant ancestors, a virus invaded the genome, but that virus, instead of doing damage, was somehow, by some unknown mechanism, repurposed and domesticated, put to new uses to serve as a gene that serves in memory. Why do that? Well, these little clumps that HIV makes, you can see them here with that arrow pointing to it, actually helps the genetic material of the virus move from cell to cell. It's kind of a capsule that protects it. That's exactly what those clumps in ARC are doing. They're protecting the genetic material as it goes from cell to cell as animals make memories. Truly remarkable stuff. In fact, our relationship with viruses is incredibly complicated. When you look at the human genome, it turns out that 8% of our entire genome is ancient viruses that are lying in our genome, defunct, dead, no longer active. Our genome is a graveyard for ancient viruses and ancient viral infections. That's really remarkable because when you look at our genome, only 2% of our genome are our own genes. We have 4 times more viral genetic material inside our genome than our own genes. And that's just the tip of the iceberg of surprises. We have so many surprises in the genome, many of which relate to evolution.